Okay, well, welcome everyone to the Biophysical Society celebration of 50 years of the Protein Data Bank. Obviously, the PDB has had a huge impact on all of our science, and it's really great to be able to celebrate the success and the impact that they've had over the past 50 years. I want to thank, to start, Francis Siporovich and Kathy Royer for conceiving this symposium, and the Biophysical Society office, particularly Jennifer Fraser, for all the great work that she's done. And I want to mention also that the symposium is being recorded, so you'll be able to get copies of it later. So I just want to give a few minutes of introduction, which is that this is sort of thing I present in an introductory biology or introductory biochemistry class, and in particular the roles of transcription and translation in biology. But we have to recognize, or we do recognize, that in fact it's not just the sequence that nature acts on. The phenotype is determined by the three-dimensional structures of the biomolecules. And the way that we have the information we have for three-dimensional structures is primarily through experimental structure determination, X-ray crystallography, NMR spectroscopy, cryo-EM. And the knowledge base of that is the PDB. So the PDB basically, in my view, is at the same level of importance in biology as the collection of structures, as transcription and translation processes themselves. So it's really fundamental. And you could imagine if there were some way, the way we can translate DNA sequence to RNA sequence, or RNA sequence to protein sequence, we would be able to understand the 3D structures just from computational calculations. And this was a vision, actually, of an international protein structure initiative that occurred between 2000 and around 2016. The long-range goal would be to make three-dimensional structures of proteins available just from their sequences through experiment, through modeling, leveraging experiments through modeling. And I think that the PDB actually has played a key role, the central role in coming close to achieving this. So the vision for the PDB is to sustain a freely accessible core archive of structured data and metadata for biological macromolecules for the public good, for research and education, a freely accessible resource. And the WWPDB, established, I guess, about 15 years ago, integrates activities across national efforts, nationally funded efforts, as well as efforts using other methods like NMR and electron microscopy. And I think both Professor Berman and Burley will actually give some more details about that. I actually wanted to just cite this one quick result, one important result. CAS, the Critical Assessment of Protein Structure Prediction, has been going on for about 25 years. And these are plots of the accuracy, the GDT, it's kind of analogous to RMSD, versus difficulty of targets over various years of CAS, and the lines are the best fits to the results. So easy targets, even early on, you could get GDT scores over 80%, meaning about 2 angstroms RMSD to the correct structure. But more difficult targets have been just that, more difficult. And basically the predictions just look like they had no basis in reality until the introduction of multiple sequence alignment methods, covariance analysis, and then particularly in the last years, using the protein data bank, the information and knowledge in the protein data bank, the structures, to learn from those structures through machine learning, how to actually accurately model structure. So here's an example of one from the recent CAS section, NMR structure, something like a 1.2 angstrom RMSD backbone between the purely predicted structure and the structure determined experimentally. And what you could see here, these open circles are the results of 96 targets. And for the most part, they're all above 80%. The average is about 90%, about 1 angstrom RMSD. So it's a very exciting result based upon the knowledge and information that comes from the experimental structures. So the current challenges which we have and we'll be talking about is what more can we learn for the PDB, and how can we learn from the PDB? how to go from these structures to their biochemical functions and maybe even cellular functions. It was a vision of the Protein Structure Initiative, which is still an ongoing activity, and we'll hear some talks about that. Dealing with multi-chain complexes and integral membrane proteins, which are particularly challenging because of how do you reconstitute them in biologically relevant forms. And so advances in understanding channels, and we'll hear a whole session on that. Molecular recognition and rational drug design. So there's an example in the upper right there of a docking based purely on knowledge from that we've learned about potential functions of a drug that binds a hepatitis C virus protease into the SARS coronavirus protease. And one docking there is the prediction, and the other is the experimental structure determined many months after. And you can see the advances that we have in being able to predict and generate structures, but still molecular recognition and rational drug design is a challenge. Also, de novo protein design with its applications both to medicine and materials. And the vision of tomography to actually take structural biology to cell biology, to actually see and build structures of whole multi-protein complexes and even organelles in the cell. And also we'll hear about this concept that, of course, structures are not static, structures are dynamic. In the lower right, you see an example of one PDB entry, but it has two different conformations determined from that structure that interconvert with thermal energy. That's actually a dengue virus protease bound to a potential inhibitor. So it's not a single structure, but it's a distribution of structures. It's just a morph. And on the left, it shows the concept that proteins live on an energy surface. And so this is kind of a vision for the future. So with that, I'd like to introduce the first speaker. So this is our session. We have a morning session on enabling understanding of protein structure, function, and design. And then there'll be a second session after the break on membrane proteins and channels. And our first speaker is Helen Berman, who is a Board of Governors Chair and Professor of Chemistry Emeritus at Rutgers University. She was also the director of the Protein Data Bank for about 20 years and really brought it very much up to the visions that we have today.